Hi, everyone. I'm Rick Fair, CEO of Bellicum. It's great to be here. Thanks to the Alliance for having us. Here to give you an introduction to our platform and our programs, and to try and do that in 15 minutes, I've had to cut out a fair amount of data, so I look forward to sharing what I can and ask you that you follow up on our website. We have a more fulsome presentation along with a number of publications that are of interest. I'll be making forward-looking statements. These statements have risk. I encourage you to look at our SEC filings for more information about those. Bellicum is a platform company. We've developed a platform we call GOCAR, which is intended to break through the limitations of current generation cell therapies. The objectives of the platform are to enhance the proliferation and functional persistence of immune cells, hopefully to enhance the power of these therapies, make them more efficacious, and because that efficacy can also be toxic, improve the performance of these cell therapies by allowing a control mechanism through a molecular switch platform that's some of the founding science of our company. GOCAR is differentiated from what you might be familiar with CAR-T cells. I'm sure I don't have to tell this audience what first and second generation CAR-T cells are and how they work. On the right, you see GOCAR design and how it differs. So we have a first generation CAR tethered to CD3 zeta signaling, and we've separated the co-stimulatory or co-activation molecules from the CAR itself. That's one differentiating design feature. We've done that so we can control when and how often cells are activated with a small molecule remideside, which leads to chemical induction of dimerization and leads to signaling. So that allows us to control frequency of activation of these cells and allows us to manage potential toxicities. The second difference from second generation CAR-T cells are the activating molecules we've chosen. MyD88 is an adapter protein in the toll-like receptor pathway. CD40 is a co-stimulatory protein. And the combination of these two molecules in our preclinical experiments led to more proliferative T cells that survive longer and that resist exhaustion better than second-generation CAR-T. It was with those preclinical findings that we chose to pursue solid tumor experiments because we believe that resistance of exhaustion and immune suppression would differentiate us more from second-generation CAR-T cells than they would in a heme malignancy. The other bit of our platform has to do with safety. CaspaCIDe, or inducible caspase-9, is the same chemical induction of dimerization technology, but instead of triggering activating signals, we're triggering caspase-9, which leads to an apoptosis cascade. The application for CaspaCIDe is really as an as-needed basis for high-grade adverse events that aren't manageable with current standards of care. So imagine a grade 4 CRS that's resistant to tocilizumab and corticosteroids, grade 4 ICANS that's resistant to corticosteroids, or in phase 1 development, perhaps an on-target off-tumor effect that's quite serious and needs quick resolution. I'll share some clinical data about CaspaCIDe, but we're highly confident this will eliminate the bulk of the genetically modified cells within hours and resolve even high-grade adverse events very quickly. We've put both of these switches into one cell now. Our second-generation CAR-T cells are what we call dual-switch GOCAR-T cells. We use remideside, our proprietary small molecule, to activate cells via IMC. And we use an approved therapy, temsirolimus, to activate CaspaCIDe, a modified version of CaspaCIDe, when needed. We've incorporated the second-generation platform into our second program, which I'll talk about, BPX-603, targeting HER2. We have two products in the clinic in phase 1/2 trials demonstrating proof of concept, both safety and preliminary activity. BPX-601 is a first-generation GOCAR-T cell, including IMC. It targets prostate stem cell antigen. And we've studied it in phase 1 trial in pancreatic and metastatic prostate cancers. BPX-603, as mentioned, is a second-generation dual-switch GOCAR-T cell, and we're studying in a basket of solid tumors expressing HER2. BPX-601, as I mentioned, targets prostate stem cell antigen. PSCA is expressed on a number of GI and GU tumors, and not so much on healthy tissues, although there is some reported expression in the GI tract and GU tract. What we've observed so far in our phase 1 studies, I should say we initiated cell dose escalation and lymphodepletion optimization in pancreatic cancer patients. We treated 24 patients with pancreatic cancer. What we observed was manageable toxicity profile, preliminary evidence of clinical activity, disease control rate of about 65%, tumor regression in 3 of the 24 patients treated, but not clinically meaningful efficacy. We pivoted subsequently to prostate cancer, where most patients, somewhere between 75 and 90% of patients, express the target PSCA. And last December, in conjunction with a private placement, we announced the results of the first 3 prostate cancer patients, which I'll share here today. As I mentioned, 3 patients were treated with 5 million cells per kilogram of BPX-601 on day 0, followed by a single dose of Remidesib to activate those cells on day 7, and then they were followed with no subsequent treatment. This is not the optimized design of IMC. These cells were designed to be reactivated on a periodic basis over time, and subsequent cohorts in the study will treat these patients weekly until either disease progression or dose-limiting toxicity. But as a nod towards safety, we wanted to evaluate preliminarily how patients would respond to the cells themselves and to a single dose of Remidesib. One of the 3 patients had a confirmed partial response. You see here the PSA response over time in this patient. They received standard Flu/Cy conditioning over 3 days. They received BPX-601 cells on day 0, and again, Remidesib at day 7. And it's really upon the administration of Remidesib, you see a very rapid and deep decline in PSA, a biomarker of prostate cancer burden, greater than 95% response in this patient. You see on the right plotting of that PSA curve with vector copy number, or CAR-T cells in circulation. And you see a pattern that we've observed in all of our clinical trials, expansion of the CAR-T cells themselves over the first few days into an empty lymphodepleted T-cell compartment, a temporal dip upon administration of Remidesib, certainly evidence of CAR T-cell activation and margination to vessel wall and into tissue, and then ultimately a rebound within a couple of days back into circulation of those CAR T-cells, and that correlated very nicely with the PSA response that you see here. Patient also had a confirmed partial response on scan, so greater than 50% reduction in the target lesion in liver. This is very, very encouraging. Again, probably not a great patient to evaluate durability of therapy because we didn't reactivate his cells after day 7, but certainly indicates that we're in the right setting to evaluate activity, and so we continue dose escalation. We saw great evidence of Remidesib-induced CAR T-cell activation, and this shows a couple of canonical cytokines associated with T-cell activation. You see in 2 of the 3 patients here comparison. In both cases, you see Remidesib-induced T-cell activation and production of cytokines, but in the patient who responded, a much larger magnitude of effect. So certainly indicative that these CAR T-cells were seeing both tumor target and getting signal 1 from the first generation CAR T-cell, or CAR T itself, and signal 2 from Remidesib, and that leads to maximum activation and cytokine production. The other important note here is that what goes up comes down. And this is a really important safety feature of our therapy. We believe that production of cytokines and chemokines is useful and important to attracting and activating a T-cell response within the tumor microenvironment. But of course, too much cytokine production that's out of control can be toxic. And with our platform, we see a great upregulation of cytokines when their cells are activated, but within a couple of days, a return to a more normal level. And that's encouraging, and it gives us hope that we'll be able to tolerate this therapy over a longer period of time. From here in the prostate cancer study, we intend to dose escalate Remidesib on a 5 million cell per kilogram flat cell therapy dose. Rationale for that you see from one of our animal studies listed here. What we see with increasing exposures to Remidesib, we see better CAR T-cell persistence, better production of inflammatory cytokines, and better anti-tumor effect. So our study design will double the dose twice of Remidesib, and we expect to report results on our dose escalation in progress early next year in the first quarter. Turn briefly to BPX-603. It's our second product candidate. BPX-603, again, is a dual switch second generation GOCAR-T cell targeting HER2. I don't have to tell you about HER2. You know it's a very good tumor target. And what you may not know is that it's been well studied with CAR T cells in several academic centers, about 100 patients treated globally with HER2-directed CAR T cells, almost exclusively second generation CAR T cells. So it gives us a nice historic control to compare to. What's been observed there is generally manageable safety, although there have been some reports of on-target off-tumor toxicity in the lung, but also a limited activity, about 8 to 10% overall response rate. So we designed BPX-603 rationally to address the limitations of those second-generation CAR-T efforts. First, about binder selection, we selected a moderate affinity SCFE and designed to optimize the trade-off between efficacy and safety. We incorporated our IMC activation switch, which I've already indicated should lead to a CAR T cell that's more potent and more resistant to exhaustion in the tumor microenvironment. And we, of course, have tethered it to our fully controllable dual switch platform, which should allow us to manage any unmanageable toxicities that occur. We announced as part of our private placement last December that we had cleared dose level 1. Given the potential safety risks of HER2-directed CAR T cell, we started an extremely low cell dose of 100,000 cells per kilogram in that dose. So we reported that we did not observe any activity nor any dose-limiting toxicity, and cell dose escalation is ongoing. We intend to present an update on this phase 1 study mid-next year. Lastly, I'll talk briefly about the CaspaCIDe safety system. Again, that's incorporated in BPX-603, but is also available to license to other companies who are developing cell therapies. We think this is a safety feature that really should be included in every cell therapy. Every car has a safety belt and an airbag. You hope you don't use them, but occasionally you need to, and when you need to, you're glad you have them. CaspaCIDe has been well studied in the preclinical setting. I didn't share that here today, but you can find publications on our website. Share a couple of clinical data sets here in summary form. One data set comes from a previous Bellicum program, RevoCell. This was a donor T-cell product added back to stem cell transplant patients, incorporating our safety switch with the rationale that if those T-cells caused graft versus host disease, which is common in the DLI setting in stem cell transplantation patients, that we could trigger the safety switch to moderate that or mitigate that. In 24 pediatric haplo-transplant patients who received donor T-cells incorporating ICAS-BASE9, who experienced steroid refractory GVHD, they received Remidicid. What we saw was a 70% overall response rate. The median time to response was at the first assessment, which was at day 1, but looking at the immune monitoring data, we saw that the majority of the reduction T-cells occurred in the first 4 hours in these patients. So a really nice outcome and great proof of concept that we can eliminate adverse event causing T-cells, and that leads to a moderation of the adverse events themselves. Last year, we haven't actually used CaspaCIDe in our CAR-T programs, but we've allowed academic centers to use CaspaCIDe as part of their cell therapy efforts, and we've started to see a data flow from those IST studies. Last year in blood, the group at North Carolina, which had a IC9 containing CD19 CAR T cell, published a case report. It's a very, very nice case report. This 26-year-old B-cell ALL patient received these CD19 CAR T cells, developed CRS early, and about a week later developed ICANS. The CRS responded to tocilizumab and steroids. The ICANS did not. So by day 10, this patient was in trouble. It was grade 4 ICANS, comatose, and they gave her Remidicid. Within 12 hours, that ICANS resolved from grade 4 to grade 1. It resolved completely within 72 hours, and she did well enough that she was asking for more CAR T cells to convert her partial response into a potentially complete response. The group at North Carolina has treated a number of other patients with Remidicid who've experienced high-grade adverse events, and we'll report on those patients at ASH this year, so we'll have more data to share. In short, this is a really great safety system. We certainly will incorporate it to all of our future products, and for those of you who are developing cell therapies who are interested, it is available for license. We currently have 7 license programs and would welcome the opportunity to partner further. I mentioned our data timelines. I don't need to reiterate them, but the first half next year will be very important to us. We'll be presenting updates on both of our studies. And with that, I will close and say that we're funded through the middle of next year. We have time to generate the data, and we think this platform merits further development, and we're optimistic that the investment community will agree. Thanks very much for your attention. I appreciate it.